ABCD syndrome. ABCD syndrome is the acronym for albinism, black clot, cell migration disorder of the neurocytes of the gut, and sensorineural deafness. It has been found to be caused by mutation in the endothelin B receptor gene. ABCD syndrome is defined as albinism, black clot, cell migration disorder of the neurocytes of the gut, and deafness. It was initially misdiagnosed and later discovered that a homozygous mutation in the EDNRB gene causes ABCD syndrome. This helped scientists discover that it is the same as type 4 Waardenburg syndrome, also known as SHA. Waardenburg syndrome. In the beginning, medical officials defined ABCD syndrome by the 4 key characteristics of the syndrome. In the first case study of a Kurdish girl, researchers described her as having albinism and a black clot at the right temporo-occipital region along Blaschko lines. Her eyelashes and brows were white, the irises in her eyes appeared to be blue, she had spots of retinal depigmentation, and she did not react to noise. The albinism is interesting in this diagnosis because the skin of an affected individual is albino pale besides the brown patches of mispigmented skin. The black clots described and seen in clinical pictures of the infants are thick patches of black hair above the ears that form a half circle reaching to the other ear to make a crest shape. As identified in this first case study and stated in a dictionary of dermatologic syndromes, ABCD syndrome has many notable features, including snow-white hair in patches, distinct black clots of hair, skin white except brown macules, deafness, irises gray to blue, nystagmus, photophobia, poor visual activity, normal melanocytes in pigmented hair and skin, and absent melanocytes in areas of leukoderma. Individuals have the blue-gray irises typical of people affected by blindness. The C of ABCD syndrome is what distinguishes this genetic disorder from BADS, and it involves cell migration disorder of the neurocytes of the gut. This characteristic occurs when nerve cells did not function correctly in the gut, which results in aganglionosis, the intestine's failure to move food along the digestive tract. Deafness or being unresponsive to noise due to very low quality of hearing was reported in every case of ABCD syndrome. The characteristics of ABCD syndrome are clearly evident in an inflicted individual. No longer considered a separate syndrome, ABCD syndrome is today considered to be a variation of SHA. Waardenburg type 4. Waardenburg syndrome is described as the combination of sensorineural hearing loss, hypopigmentation of skin and hair, and pigmentary disturbances of the irids. Hearing loss and deafness, skin mispigmentation and albinism, and pigmentary changes in irises are the similarities between WS and ABCD. According to a dictionary of dermatologic syndromes, Waardenburg syndrome has many notable features, including depigmentation of hair and skin, white forelock and premature graying of hair, confluent thick eyebrows, heterochromic irids or hypopigmentation of iris, laterally displaced inner canthi, congenital sensorineural deafness, broad nasal root, autosomal dominant disorder, and other associated findings, including black forelocks. Researchers in the past 20 years have determined that a gene mutation, specifically a homozygous mutation in the EDNRB gene, is the cause of ABCD syndrome. The advancement of technology led to new DNA material testing methods, and this discovery changed the view of ABCD syndrome completely. A homozygous mutation means that there was an identical mutation on both the maternal and paternal genes. The identifying clinical report stated the test was done by scanning the Kurdish family for mutations in the EDNRB gene and the EDN3 gene by using a test called denaturing gradient gel electrophoresis. The electrophoresis test takes advantage of electrical currents and differences in melting points of fragments of DNA or RNA to move them based on their molecular weight. The differences in mobility of the fragments then can be analyzed to determine different sequences and to detect individual alleles. Different nucleotides in DNA are codes for certain proteins, which are formed by different patterns of the base pairs adenine, thymine, guanine, and cytosine. The combination of adenine and thymine and guanine and cytosine align on the double strands of DNA. The test results found an aberrant DGGE pattern of exon 3 of the adenine gene. The mutation was determined to be a homozygous C to T base pair transition at the amino acid level, causing a premature stop in gene translation. This specialized testing enables geneticists to recognize the gene mutation that is the cause of ABCD syndrome. New findings introduced an important break in the beliefs about ABCD syndrome because the endothelin B gene is a gene involved in Wahr-Ordenberg syndrome. The endothelin receptor B produces Wahr-Ordenberg syndrome type 4. Researchers began discussing the possibility that ABCD syndrome was in fact not a syndrome, rather it was a type of another syndrome known as Wahr-Ordenberg. Discovering that the same gene is involved in ABCD and Wahr-Ordenberg syndrome is important because researchers can now look further into ways to fix this crucial gene. The occurrence of WS has been reported to be 1 in 45,000 in Europe. The diagnosis can be made prenatally by ultrasound due to the phenotype displaying pigmentary disturbances, facial abnormalities, and other developmental defects. After birth, the diagnosis is initially made symptomatically and can be confirmed through genetic testing. If the diagnosis is not made early enough, complications can arise from Hirschsprung's disease. Screening generally only takes place among those displaying several of the symptoms of ABCD, but a study on a large group of institutionalized deaf people in Colombia revealed that 38% of them were Wahr-Ordenberg patients. Because of its rarity, none of the patients were diagnosed with ABCD. Nothing can be done to prevent the disease. Treatment for the disease itself is non-existent, but there are options for most of the symptoms. For example, one suffering from hearing loss would be given hearing aids, and those with Hirschsprung's disorder can be treated with a colostomy. If the Hirschsprung's disease is treated in time, ABCD sufferers live otherwise healthy lives. If it is not found soon enough, death often occurs in infancy. For those suffering hearing loss, it is generally regressive and the damage to hearing increases over time. Digestive problems from the colostomy and reattachment may exist, but most cases can be treated with laxatives. The only other debilitating symptom is hearing loss, which is usually degenerative and can only be treated with surgery or hearing aids. Dutch ophthalmologist Petrus Johannes Wahr-Ordenberg brought about the idea of Wahr-Ordenberg syndrome when he examined two deaf twins. Wahr-Ordenberg decided to define the syndrome with the 6 major symptoms that patients most commonly had. He defined lateral displacement of the medial canthi combined with dystopia of the lacrimal punctum and blepharophimosis, referring to people with broader and flatter nasal bridges, which in turn leads to folds in the skin that cover the inner corners of the eye. Secondly, people who are born with a prominent broad nasal root have a widened area between the eyes, causing them to have a flatter and wider face, along with eyes farther apart than normal. Thirdly, hypertrichosis of the medial part of the eyebrows is present, meaning excessive hair growth in the patient's eyebrow region, most likely leading to a unibrow. The fourth symptom, white forelock, was commonly seen as the pigmented strands of hair. Heterochromia iridis indicates that the patient has 2 different colored eyes or 2 colors in the same eyes. Deaf. Mutism. People with the disorder are both deaf and mute. When scientists further investigated the syndrome, they realized that patients exhibited a wider range of symptoms of this disease in different combinations. This helped them distinguish forms of Wahr-Ordenberg syndrome. Their evaluation consisted of specifying Wahr-Ordenberg syndrome type I, type 2, type 3, and type 4. In 1995, a case study was performed of a Kurdish family. Scientists completed a molecular analysis with DNA strands of the patients diagnosed with ABCD syndrome. Their task was to scan the sequences to find a mutation in the ADNR gene, one of the most important protein coding genes. When they completed the scan, they found a homozygous C to T transition resulting, at the amino acid level, in a premature stop codon. Then, they went back and defined that Shaw-Waardenburg syndrome consisted majorly of mutations in the NDRBR3 gene, along with SOX10 mutations. Therefore, the researchers confirmed that ABCD syndrome was a form of Shaw-Waardenburg syndrome. The genetic tests that they performed on the patient's DNA helped in identifying the appropriate diagnosis. In 2002, Woodcock and other scientists examined patients born with white hair, some black locks, and a pigmented skin. He diagnosed them as having black lock albinism deafness syndrome. Those who were closely working with this case suggested that it was an autoimmune disorder rather than a genetic defect. However, soon after, they had a patient who was one of 14 children of Kurdish parents. The pedigree they examined revealed autosomal recessive inheritance, which led to cell migration of the neurocytes in the gut and, therefore, they redefined the syndrome as ABCD syndrome. This revealed a homozygous nonsense mutation in the ADNR gene, meaning that ABCD syndrome was not a separate entity but rather the same as Shaw-Waardenburg syndrome. Thanks for watching this video. Subscribe to our YouTube channel for more videos.